Thanks, Ray, for the introduction. You, Ray, you and all the P&I people for the kind invitation to this wonderful meeting. This is really inspiring, really scientific. I'm happy to be here. Thank you for holding on. I'll try to make it short and exciting to make the short time towards the evening. So I will present you some information on our messenger RNA nanomedicines technology platform, and will give you some insight into the microfluidics technology we use. It's still work in progress, but anyway, it could be worthwhile to be mentioned. Now I look for the procedure.
Yes, there you go.
Yeah. Okay. I'm from Germany, Mainz, Germany, from a company called BioNTech, which is a spin-out from the Mainz University. We are about 20 minutes by public transportation from the Frankfurt airport. A relatively young, not so young anymore company. However, fastly growing. By now we are way above 400 employees, and we are interested in all kinds of tumor and immuno and other therapies. I show you this to a bit more detail on this slide. So BioNTech is kind of a holding with different independently acting subsidiaries, which are basically all in one way or the other acting in the fields of cancer therapy, immunotherapy, cell and gene therapy, diagnostics, and RNA-based therapies. And the RNA-based therapies is what I would like to report about. Maybe interestingly, we also have a manufacturing site, which is the one here. It's about 100 kilometers west of our city, where we can produce RNAs, liposomes, and we will produce also a Lipoplex product soon for our next clinical study. Here, this one is a very easy, simple introduction to what this is all about. We talk about RNA nanomedicines, which can be used for different applications. For us, then the first line of interest is tumor immunotherapy. However, you can also use it for vaccination purposes, for expression of therapeutic proteins. And you have many different challenges which you have to tackle. I today would like to talk only on the formulation aspect, this upper part, and on products for tumor immunotherapy, just for sake of time. One slide just to get back very quickly to immunotherapy, immune recognition of cancer. As you probably are familiar with, tumor cells can be recognized by the immune system by tumor-associated antigens, and there will be identified, attacked, and destroyed in a similar way as our immune system attacks other pathogens like in a viral infection. So the idea is to make use of the immune system in order to combat cancer. How do we do it? We heard many different presentations in this field already. Instead of doing a conventional vaccination using antigens, we use messenger RNA, which codes for the antigens which we want to use for therapeutics. And for that, we need to bring the tumor antigen coding RNA into immune cells, into dendritic cells, which are the center point for immune responses. If this is successful, we can initiate a whole cascade of immune responses, both systemic immune responses in terms of interferon alpha and so on, and as well as specific immune responses in the sense of expansions of T cells, which specifically attack those cells which present the antigens that they are specific for. And the key technological challenge is now to bring the RNA into the target cells. As RNA is labeled, it's a delicate molecule. It's not easy to get into the cell. For that, we use nanoparticles, as obvious from this meeting here. And the technology I would like to present today is based on lipoplexes, where we use RNA and liposomes, which you incubate to each other, and we end up in a nanoparticle, a lipoplex nanoparticle, which is intravenously injectable, and which allows us to deliver the RNA specifically to certain organs or to certain compartments in the body. Yes. How do we make the formulations? It's kind of a physicochemical screening and biological evaluation process, where we start with all of our different components. Now, or really maybe use the pointer. So we have RNA, lipid carriers, namely liposomes with different properties, ionic environments, which may be used to condition either the RNA or the lipid carrier, which may be applied before, during, or after incubation of both components to each other. And we use different preparation protocols to form these particles. And in each case, we end up with in a specific particle characteristics, which here is depicted by a lamellar phase. And after this physicochemical characterization, we bring in the particles if they are suitable to biological testing. And the fundamental aim is to really come up with kind of a structure-function correlation, in the sense that we can really correlate certain physicochemical characteristics with the biological activity, with which we're really interesting. Maybe for you, Yoon, the idea for the next instrument, if you could make a nano assembler where you can program the screening, this could be really great. But so far we had to do this by hand and by scientists and by technicians, and we did it to keep the things simple with simple components. Here you see a selection of very well-known lipids, standard lipids, which you can buy off the shelf from different providers. And we were able to demonstrate that we can assemble our delivery vehicles using these standard components. So our principle of targeting is not so much one where we have a certain particular lipid which has been identified by screening of hundreds or thousands of components. It's more a particle engineering, a controlled assembly using building blocks with which we adjust certain molecular properties. And in order to do this, we used at the screening phase standard technologies. You have seen that. This is the lipid film hydration method, which enabled us to do things easily at the small scale and up with defined properties. Particularly size was interesting for us. And in the end, this is what we could show. Here you see two different formulations of messenger RNA coding for luciferase. And afterwards, we have made in vivo bioluminescence measurements. And you see, first of all, these formulations we made were able to protect the RNA from degradation in circulation. And furthermore, we get expression in certain organs where, interestingly, the expression profile is different for these two formulations. In one formulation here, for example, we have a preferential lung targeting, and the other one, a preferential spleen targeting. And the spleen for vaccination approaches is very interesting because it's the main lymphoid organ with lots of antigen-presenting cells. And therefore, it is a promising basis to develop vaccines, mRNA vaccines on this basis. Interestingly, in contrast to many other presentations here, we don't have any liver signal, and this was on purpose. Here we show you the formulations which don't display liver expression. It is one of the parameters in the formulation space, particularly by selecting the right lipid composition, by which we could more or less completely erase the expression in the liver, which does not mean that the lipids do not come to the liver. If you have identified such a principle, you can do a clinical development. I think I have to call these now transfera extruders, right, which we used in the beginning. But luckily, we were able to scale this up, also as been shown in the presentation before, using better scalable methods. This is an ethanol injection-based approach, and this is a situation in our GMP facility where we manufacture the liposomes on the basis of the protocol developed here. And to give you a short overview, it's a modified ethanol injection technique where we were able to adjust particularly the particle size just by the process parameters. We do not need any extrusion and particular processing. There's just one processing step, which is a filtration to get rid of potentially large particles, which are not there, but still we do it for safety reasons. And it's a very straightforward protocol where we don't need dialysis, cross-flow filtration, or something else to get rid of any component. We end up with particles of defined size and properties which are optimized for the optimized activity of the RNA Lipoplex product. We followed for our first clinical studies what we called a kit approach, where we manufacture the components of the lipoplexes individually and fill them individually. This has different reasons of stability, shelf life stability, of logistics, of development effort, regulatory reasons, and also reasons to enable using individual components for several clinical studies. And in the end, this was a very straightforward approach where we get our injectable medication just by adding a diluent and liposomes to the RNA. And you see here you have a slightly opaque solution while above it's clear. So that's a directly injectable product. This has now entered clinical studies for application in malignant melanoma. We apply 4 different RNA coding for 4 different tumor antigens as an intravenous injection. Yeah, okay, no, I think I missed one slide. This is now currently ongoing, where we have first results from clinics indicating both systemic and specific response on the injection in the sense of interferon alpha levels and elevated titers of the T cells specific for the antigens we have vaccinated for. On this basis, or in this context, we have set up plannings for several other clinical studies, which I have segregated here in 3 different groups. One we call FixVac. This means patients receive a fixed combination of different vaccines. This is what you have seen a minute before. Another approach is what we call warehouse, when patients are screened if they are positive for certain antigens, and then they receive medication only comprising the RNA coding for these antigens they are positive for. And then we have a strictly individualized branch where each patient gets his tumor sequenced and analyzed in order to identify antigenic mutations, and on this basis a personalized medication for this particular patient is synthesized. And at this stage, actually, we practically entered into all the microfluidic technologies for manufacturing of patient batches. As for this individualized product, we have a very small batch size, so we didn't have such a large effort for upscaling, and we were very close to our laboratory scale. and we intended to develop a process in order to provide the ready-to-use product by ideally a microfluidic process. The difference between the other two settings is that here the batch size is very small, in the order of a few hundreds of milliliters. Here the batch size is already much larger. We would like to manufacture a bit larger batches, and here this is just a generic process where we also would need conventional batch sizes as an ordinary industrial production. So we looked at microfluidic techniques already for a very long time, beginning with techniques for making liposomes with microfluidic manufacturing. This is from 2004 when we first got interested in that. This is an example where people investigated the process of condensation in situ in the channels, and all of this stimulated us for the step towards development of manufacturing of clinical batches, which should look like that, to combine RNA and liposomes in a mixing device to end up in the bulk product, which would be filled then and stored at 4 degrees, frozen or lyophilized. This is what you know, NanoSampler. We've used this quite a lot for screening and development at lab scale. Here you see some custom-made in-house setups with syringe pumps, with different types of mixers, very large ones and very small ones from providers which were in the local environment. And to sum this up, I would like to show you some results which we obtained from the NanoSampler, where in simple words we were able to show a very nice coherence between the hand-mixed and the microfluidic mixed particles. We got same physicochemical and same biological properties, as you see here from the size measurements, from the luciferase expression in vitro, and from the in vivo bioluminescence measurement. However, at this stage, we encountered some technological problems in the sense that aggregation and clogging was observed, so we have to go another round in fundamental development to set up mixers where we avoid these problems of clogging and aggregation in order to scale this up. And for the time being, we work with a simplified protocol in order to maintain clinical development. With that, as I see, I'm already late, and I don't want to delay this further on. I would like to thank my collaborators. I would like to thank you. And if you are still strong enough, I'm happy to take questions.
Thanks, Heinrich. It's amazing to see the work that you're doing with BioNTech in this personalized vaccine space. I'm sure a lot of people have questions on the regulatory challenges that you may face or have faced trying to bring this to the clinic. So maybe you can talk a little bit about that.
Yes, I think the regulatory challenges are, of course, considerable. It depends on the type of medication. The largest, the highest challenges are in the field of this personalized medications, as we need to kind of validate and qualify a process and not a product. So it is very difficult to do the full quality control for personalized medication, which is manufactured at small scale with a very high frequency. So we plan to manufacture several medications per day, and we plan to provide this medication very soon after the patients have been screened back to the patient, because otherwise it's too late to bring it in. And we are in close contact with the local authorities, the Paul-Ehrlich Institute, where more or less this is work in progress, how to solve this problem. Major problem, for example, is sterility. So you have to test the samples for sterility, which normally takes kind of 4 weeks. And the patients do not want to wait 4 weeks for the product to be stable. So it means we have to set up a process which intrinsically shows a very low risk of introducing instability, where maybe in a pre-final step one tests already in order to be a little bit faster, and where part of the medications or the medications can already go to the patient so that they are treated, and the sterility testing comes later, so that you would do some intervention if there was any contamination found, just in order to reduce the time lag between manufacturing and treating the patient.
I'm going to hog this mic then and ask you another question, Heinrich. There is? Okay.
With your lung delivery, I was wondering if you can provide some insight into what cell types in the lung you think you're delivering to. Is it mostly the endothelial cells, which is known for cationic liposomes when injected intravenously?
Yes, it's to a big part endothelial cells. It depends on the type of formulation. So we can really modulate the profile of preference by changing the formulation properties. But as known in the literature, we target predominantly endothelial cells with the standard formulations.
I'm going to sneak in one very, very quick question, if I may. So with your current clinical product, you're using lipoplexes, which are, cationically charged. They're fairly big. How are you administering them? Is it IM injection?
It's intravenous injection.
Intravenous?
They're not positively charged, actually. They're negatively charged. So that's a big advantage, as you have much less risks in comparison to classical gene delivery vehicles, which are usually positively charged. But it's an intravenous injection, which is unusual for a vaccine, but it's rather usual for drug delivery person. And in principle, it has a very big advantage, as you can target a much larger compartment of lymphoid cells. So we think that it's a very good approach for bringing this really into clinical practice.
In your spleen-specific formulation, are those really APC-specific?
Yes. So we have abundant data on that. I can show you them. So we have really investigated the target cells to detail, for example, by depleting dendritic cells, and where we saw then that the fluorescent signal went down, and by a large number of other methods. This is all published in our recent Nature paper, where you find abundant data on that.
Sorry, I'm making everyone wait. Just a very basic question on the type of cancers you would like to target. Is there any? Can you comment on? You see this working better for one type than another?
In principle, this approach should be applicable in a generic manner. Should be applicable for any type of cancer. The reason why melanoma has been selected was for sure that here the tumor antigens are well known and identified. Our next indication will be mammocarcinoma, breast cancer. We discuss other types of cancer. We had already discussions with glioma, with pancreatic cancer. It depends a bit on the clinical setting, and potentially also on combination therapies you apply. But in principle, it should be generically applicable for any kind of tumor.